Okay, moving along, we're now going to hear a virtual talk by Jason Stajich on genomics of coccidioides, and if his plane lands on time, he may be around for the question and answer period. He's flying from Quebec to Montreal right now.
Hello, my name is Jason Stajich. I'm professor in the Department of Microbiology and Plant Pathology, and I'm delighted to be able to present with you some of our research on coccidioides genomics and comparative analyses. I'm sorry I can't be there with you in person, but I'm excited to interact with the session as best I can online. I want to first acknowledge and talk about some of the reasons why we're trying to tackle genomics and coccidioides. It's important to build these resources to be able to investigate some of the novel biology that coccidioides displays. We also need to better understand this genome content stability among different isolates and species to know how variable different isolates might be. And using these tools, it's important to be able to develop the gene knockout systems and other genetic manipulations. Further work that's been exploring gene expression differences between stages like spherules and mycelia have benefited from the genomic tools to be able to profile the genes that change. Also, the understanding the specific metabolism of cocci and its potential susceptibility or resistance to different antifungals, as well as other kinds of treatment options, are important that we can glean from some of the genomic studies, and of course, understanding the full complement of proteins that are produced and where they and their functions are important in some of the vaccine development work and characterizing what the biology of the organism and its makeup. So how are coccidioides genomes structured? How large are these genomes and compared to other nigonalian fungi? These have been questions that were first answered in some of the initial genome sequencing projects of cocci and its relatives. We also want to understand where, how compact the genome is. Are there genes that are dispersed throughout the genome? Are they localized in some places and mostly filled with transposable elements in a different place? In different parts of the genome, what kind of compartmentalization is there? Finally, or also, how big are gene families, and what is the gene content distribution? It's important in just understanding the basics of the organism, but also its potential to adapt to different environments. And then finally, we're particularly interested in how transposable elements shape the genome of fungi and understanding how variable and how much these proliferate in different strains or species. A reminder of the relationships among the major groups of fungi. In bold here, you can see the different phyla listed out. The nigonalian fungi that coccidioides is a member of is part of the Pizizomycetina, the filamentous ascomycetes. There are, of course, many species in these different lineages, but you can see that some of these are some of the ones that are more closely related to cocci include things like Aspergillus and Fusarium and some of the other species in those Pizizomycetina lineages. When we look more closely at the phylogeny of the nigonalian fungi, here is a sampling of species with genome sequences available. You can see cocci there in the middle with several different relatives, some of which are pathogenic, some of which are not animal pathogens. I put some emojis here that sort of generally represent some of the hosts that these fungi are found on, though not isn't as scientific completely, but you can see again that you can see where cocci is in proximity to some of the other relatives in this group. Importantly, when we try to understand the genome of cocci, we would like to understand it in context of other genomes of fungi. And so the kingdom has benefited from the extensive efforts to sequence genomes. You can see there are about 1,600 annotated published genomes in GenBank. There are actually more that are not annotated, but the distribution of sizes are here, and on the x-axis is a log scale of the size of the genome, and on the y-axis is the number of genes found in each genome. So as the genome size gets bigger, you typically see more genes because there's more real estate to put those genes in, but you do see examples of things that are off that diagonal, like the purple groups, the Entomopterales, Entomoptera fungi that have very large genomes, but not necessarily a huge number of genes, and that's driven by large numbers of transposable elements in those genomes. Coxy falls in the middle here. You can see at about 30 megabases and 10,000 genes. Some of the things that are smaller, some of the species that have smaller genomes include in bright red, those Microsporidia, which are obligate pathogens and tend to have smaller genomes and many fewer genes. When we look at Coxy in context of the other Inigonalian genomes, we can see, again, it falls maybe at the upper end of that in terms of number of genes, but it's really kind of not an outstanding in any way relative to the other species. There's a distribution here you can see of sizes that they're all in approximately the same ballpark. What genomic resources have already been generated for Coxy and are available? These include some of the early work that this foundational work from the Fungal Genome Initiative led through the Broad Institute, as well as efforts at Tiger, which is now JCVI, that generated Stanger genome sequences for 2 of the species, and then additional Sanger genomes that were more draft quality from different isolates within the 2 species. These are really important starting points for so much of the biological investigations I mentioned. There's also been population sequencing that's been completed from Illumina, using Illumina sequencing, and that's been through a consortia of different groups, both Arizona and CDC, TGen, NAU, and additional other contributions. This is important for measuring the diversity within the species as well. Of course, there's been transcriptional profiling through RNA-seq and microarray work, and then some additional reference genomes with long-read sequencing that have been generated, including a new genome of Posadiceae, and some that's published, and some ongoing work that our lab is participating in collaboration with Mark Orbach, using PacBio in several strains. So first, the Silviera genome, Coccidioides Posadiceae silviera, was published earlier this year, led by Bridget Barker's lab, and that included this new assembly that had the genome in basically 5 scaffolds, which represent 5 chromosomes. You can see here on the right these dot plots of sort of the diversity or the repetitive element content potentially in each of these chromosomes, as well as just a depiction of the genome size, of the scaffold sizes. But additional work that's been going on since then in Mark's lab has generated some additional genomes with the HiFi PacBio sequencing technology, and you can see here some of the top 4 of these new genomes, and then the Posadiceae from earlier this year is listed. And so again, we see genome sizes a little bit less than 30 megabases. For some, the scaffold count, again, represents all the scaffolds that come out of the assembly, but almost all of the sequences in the first 5 chromosomes. And in cases when we start to look for the presence of telomeric repeats, we can find at least 5 of the scaffolds have telomeric repeats for these 4 genomes I'm mentioning here. But in addition, several of these strains assemblies have telomere to telomere assemblies where there's a telomere on both ends of the scaffold, indicating we have a complete chromosome put together. So that's pretty exciting to be able to generate this directly from the HiFi sequencing alone. When we go in and predict genes, we also have sort of relatively similar gene content, although there's variation, and that can be driven both by the way that the gene predictors work, the possibility of some sequencing errors causing genes to be split up, and also the true biological differences between strains. And so we're still on the hunt to understand the full breadth of what those differences mean. And then you can also see that the repetitive content varies quite a lot between these strains, and in particular in between species. And so that's definitely further work to understand when and how that those differences arose, and their importance. When we look at these repetitive elements, we can draw these landscape repeat graphs. At the top, you can see Coxy Posadiceae, one of the strains, and then we have Coxy imidis at the bottom. These landscape plots show a diversity of the transposable elements, and so the x-axis is a diversity measurement, and so all of the elements that are part of a family that are within a certain distance of each other are binned in one of these bins here. And so if they're at a bin 5, and that represents sort of an average diversity difference of a Camorra distance of 5. And so this helps us get a sense of whether or not the transposons are a constant process where you're always getting new elements, and that would mean that you would see a lot of elements kind of at the left-hand side here where there's very little divergence, because if they're recently transposed, there should be another copy of itself, so it should be very similar. But if a lot of the transposition occurred a while ago and the copies have just diverged, then we'll start to see these humps at a little bit later age. And so that's what we're seeing more in imidis. We're seeing a lot of diversity at around this 15 time point, 15 scale, and that represents perhaps a bunch of transposition that occurred at one point in time. And so there's a lot of elements of the same age. But they have diverged from each other. So again, just thinking about the timing of when some of these changes occurred is what the goal of those graphs. There is this big peak at 0 in the post-Odysseia data set, which most of those elements are all in gray, so we don't know. We haven't come up with an identification of them. Possible that's an artifact. It's something we still need to really be sure of, but that is what the data represents right now. You can also see that about 82% of the genome is not repetitive, and again, so most of the genome is genic, but there are these major classes of LTR elements as the most dominant types in both species, and that's pretty common across most fungi too. So when we look at the genome across different isolates, the question is, how plastic are those genomes? How much variability is there? And so using published data sets that were generated through the CDC and NAU and TGen, we see we've had sequencing of both imidis and post-Odysseia populations, and I went in and called sequence single nucleotide polymorphism variants in these genomes, and then built a phylogenetic tree from those to be able to represent diversity, or to see them in their diversity. So then we use those same sequencing reads that were used in the sequencing to call SNPs, and we look at where those reads map in the genome. We map them back to the reference genome, and we then we calculate the average depth of coverage across a particular genome. So here I'm just showing you post-Odysseia chromosome 1 and imidis chromosome 1, and the top track is this normalized depth of coverage. Because you don't sequence the same number of reads in every experiment, we normalize the depth of coverage for that, and so we normalize it to 1. We expect about an average, the way this is normalized, to 1, and then when there's extra sequence, you'll see this as a plot where the values are above 1. If something is missing, then it'll go below 1. So if something is at 2, then represents something that might represent a ploidy difference or an extra copy of a particular region of the genome. So the search for copy number variants, and this is also a similar approach that's used in Saccharomyces and Candida to look for drug resistance aneuploidies, as well as copy number variation that's seen in cancer cell lines and cancer cells. And so we're adopting the same kind of strategy. One thing you can see here in these other two tracks that represent repeats and gene density is, of course, that they are anti-correlated, because we typically mask a region and don't predict genes in the region where there are repeats. But you can see that a lot of these variable regions within the genome correlate with the repetitive regions. And so some of these places with lots of sort of spiky spots are kind of islands of many transposable element repeats. Likely the telomeres often are places where you accumulate these, as well as probably this region in the middle that has a high density is probably the centromere. But I highlighted in this orange box a couple of regions on chromosome 1 where you can see a green color there that represents a higher coverage than 1, and that likely represents some sort of copy number variation that's only found in some of the strains. Each strain is plotted with a different color, and so you can see only the green strain in this particular plot shows that higher coverage. If we zoom in, if we zoom in to just post-Odysseia for a second here, and I've now unfortunately, or confusingly drawn the whole chromosome now. So the whole genome. So this is chromosome 1 here in this orange box, but then we also have chromosome 2 and 3 and 4 further along. But you can see in this zoomed in region of this orange box where there is some extra coverage for the post-Odysseia genome chromosome 1. And so, but you can see the colors for the strains here. In one case, there's an orange and a yellow, and that represents a place in the genome where there's extra coverage. And if we zoom in even further and just look at that chromosome 1 again, you can see that when we identify the colors to strains, we're able to tell that their particular strain has, this is GT162, has this extra coverage in this region. And so this is a candidate for further detailed look for copy number variation and what genes might underlie it. You can also see a yellow block here for coming from the J. Taurus strain, and then starting up on chromosome 2, there's a big block on from the GT2 Texas strain. So this kind of investigation allows us a way to start to determine if there are copy number variations and potentially what kinds of genes might be found in there. So how do we do a better job of trying to figure out the functional parts or investigate these gene regions? It's important to have database resources. An important one like that is FungiDB, and I'm showing you here a couple of screenshots of a genome browser that allows you to investigate a region of the genome and start to look at the genes and dynamically zoom in and zoom out. You can also build queries up in FungiDB. Here's an example query that's not really asking a question about the CNV project, but where we've taken a query for all the proteins that are signal peptides that are likely to be secreted, and then overlapped that or intersected that with a set of genes that were predicted or identified to be upregulated in the spherules phase. And so the intersection between those is only 147 genes, and we can then look at that list of genes, we can sort it by expression levels and try to identify what those genes might be. And so this first top gene is this proline-rich antigen. If we click on that gene and get a gene page view of it, we can start to, actually I'm looking at the second one, sorry. So it's a hypothetical protein. We don't even know anything about this gene. When I zoom in and look at this gene, I can see homologs to this gene in other species and their predicted functions as well as conserved domains. So FungiDB provides a nice interface for that where you can start to investigate those. Finally, just a little bit of data advertisement of a paper that was published earlier this year from Aaron Carlin's lab at UCSD. This includes a comparison between using this new technique called csRNA-seq for cap small RNA sequencing. And in this case, there's a contrast between mycelial and parasitic phase work, and the technique is able to capture these actively initiated transcripts, and that's how it provides this very specific, precise resolution to where transcription is initiating. And using this technique can start to examine the landscape of where genes are being transcribed during these developmental stages and start to understand potentially the control of that expression. And so we've got these basically twofold more number of these sites in spheruleal phase than we do in the mycelial phase. And so that's a really exciting result. I'm only doing a very high view of this results in this paper. I would commend your taking a deeper look at it if you're interested. But this work is able to define these promoter regions much more accurately to this very high resolution, and identify the potential that there's additional or alternative regulation besides just sort of cis-regulatory motifs in controlling transcriptional regulation in this transition from mycelial to spheruleal. Again, having these genomic resources and these browsers and these tools are all critical to being able to investigate and start to build more functional interpretation. So in closing, thanks so much to the folks that have participated in the work. Kelsey is a postdoc in my group, working on the Coxy genomes and their assembly and annotation. That's in collaboration with Mark Orbach's lab, who generated the sequence data and has been a collaborator throughout. And also the other work that I've talked about with Silviera has been, and other projects, has been a long-term collaboration with Bridget Barker's lab, as well as with Marcus working with her. And it's been a fantastic collaboration over the years. And also acknowledging the work that with Sasha and his work and in Aaron's lab and others at UCSD who contributed and are intellectually to this really exciting new approach to understanding transcriptional regulation in Coxy. And finally, the work that we are supported, we're supported through funding that in the UC Valley Fever Consortium that Anita has led, and really appreciative of all of the leadership and the funding that allows us to dig deep into this work. With that, thanks so much for the chance to speak, and I look forward to interacting with you in questions in other formats, even if I can't do it directly at the meeting at this moment. Thank you.